Welcome to part 2 of the chapter 3 lecture on cell biology for anatomy and physiology 1. So we left off talking about all of the various types of organelles. Right? Previously we finished up talking about the Golgi complex. So now we're going to pick up with the lysosome. Right? And this is an important sort of little organelle because this is going to be really important, and it's going to contain a lot of really powerful digestive enzymes. So the lysosomes are going to be membrane-enclosed vesicles that are going to form in the Golgi complex. And like we said, they're going to contain those digestive enzymes. Lysosomes are going to be involved in digesting substances that enter the cell via endocytosis. And they're also going to transport final products of digestion into the cytosol. Lysosomes are going to carry out autophagy, and this is going to be the digestion of worn-out cells. And they're also going to implement autolysis, when this is going to be the digestion of the entire cell. Lysosomes are also going to accomplish any kind of extracellular digestion that needs to occur. Next, we have the peroxisome. These are going to be structures that are similar in shape to the lysosome, but they are going to be a little bit smaller, and they are going to contain enzymes that use oxygen to oxidize or break down any kind of organic substances. And then we have the proteasome, and these are going to be barrel-shaped structures that are going to be there to destroy any sort of unneeded, damaged or worn out or faulty proteins, and they do this by cutting those long proteins into small little peptide chunks. So you can see our proteasome. Next, we have the mitochondria. And every American kid who's ever gone to school at all in the States has heard that the mitochondria is the powerhouse of the cell. Now, this, for whatever reason, is very distinctly American. Apparently, other countries don't teach it quite like this. But the reason that we know the mitochondria to be the powerhouse of the cell is because its primary role is to be the generation of ATP, and it does this via aerobic respiration. This just means that it requires oxygen. So active cells that use ATP in high rates, things that are found in like the muscles or in the kidneys or in the livers, things that are going to, or organs that are going to be or tissues that are very metabolically active, are going to have very large numbers of mitochondria. So they're going to have a lot more mitochondria so that they can keep up with the energy demands of that cell or that tissue. So a mitochondria is going to consist of an external mitochondrial membrane and an internal mitochondrial membrane, and there's also going to be a small fluid-filled space that's in between them. So both membranes are going to be similar in structure to that of the plasma membrane, and the internal mitochondrial membrane is going to contain a series of folds, and these are called mitochondrial cristae. You can see them here, our series of folds in our inner mitochondrial membrane there. The central fluid-filled cavity of the mitochondrion is going to be enclosed by the internal mitochondrial membrane, and this is called the mitochondrial matrix. You can see this here. The elaborate folds of the cristae, or of the mitochondria, are going to provide a huge amount of surface area that's going to allow for chemical reactions to occur that are all going to be part of the aerobic phase of cellular respiration. And this is the respiration that's responsible for producing most of the cell's ATP, or most of the energy for the cell. So this is why it gets the name as of the powerhouse of the cell, because it's responsible for producing most of the ATP that the cell needs. Mitochondria are also going to, interestingly, play a very important role in apoptosis. So apoptosis is essentially scheduled cell death. So this is going to be in response to any kind of stimuli that is potentially harmful to the cell. Things like a large number of destructive free radicals, so like reactive oxygen species, as well as any kind of DNA damage, growth factor deprivation, or a lack of oxygen, hypoxia, as well as a lack of nutrients. Also, certain types of chemicals. Exposure can cause apoptosis to occur. So certain chemicals are going to be released from the mitochondria that follow the formation of a pore in the outer mitochondrial membrane. So one of the chemicals that's being released from the cytosol or into the cytosol of the cell is called cytochrome C. And when it's inside the mitochondria, this is going to be a really important enzyme or chemical in the whole process of aerobic cellular respiration. However, once that pore forms in our outer mitochondrial membrane and cytochrome C enters the cytosol, this and other substances are going to initiate a cascade of activity or of activation of protein digesting enzymes that's going to bring about this scheduled cell death, this apoptosis. Another important fact about mitochondria is that they are self-replicating and that mitochondrial genes are inherited only from your mother. So the DNA that you have that's going to make up the mitochondria is unique to your mitochondria, and it is inherited exclusively from your mother. So that's the last of the organelles that we're really going to talk about. We're going to now move on to the nucleus, and this is going to be a really important part of the cell. So most of the cells in the body are going to have a single nucleus. There are a couple of oddballs, things like red blood cells that don't have any nuclei. They are anucleated. And then there are others that have multiple nuclei. These are multinucleated. This is things like skeletal muscle fibers that are going to be multinucleated. So functionally, the nucleus is going to control cellular structures. It's going to direct cellular activities, and it's also going to produce ribosomes in the nucleoli, which we'll talk about in just a minute. So the part of the nucleus is going to have several parts. There's going to be a double membrane called the nuclear envelope, and this is going to separate the nucleus from the cytoplasm. So both layers of the nuclear envelope are going to be lipid bilayers, and this is going to be very similar in structure to what we see for the plasma membrane. There are also going to be many openings called nuclear pores that are going to extend through that nuclear envelope, and each nuclear pore is going to consist of a circular arrangement of proteins that are going to be around a large central opening. You can kind of see that here. Around a large central opening, and this is going to control the movement of substances into and out of the nucleus, or between the nucleus and the cytoplasm. Then within that nucleus also, you can find one or more of these little spherical bodies called nucleoli, and this was sort of my specialty in graduate school. It's these nucleoli that are going to function in producing ribosomes. So the main function of the nucleoli within that nucleus is going to be ribosome biogenesis. So within the nucleus are going to be these, within the nucleus of most of these cells, this is where we're going to find the cell's hereditary units, and these are called genes. So these are going to control all of the cellular structures and are going to have basically the directions, the blueprints to direct any kind of cellular structure or cellular activity for that specific cell. Within that nucleus, like we said, we've got genes, and these are going to be arranged along chromosomes. So humans are going to be have somatic cells. This means that they have 46 chromosomes, and therefore 23 are going to be inherited from each of the parents. 23 from mom, 23 from dad. So each chromosome is going to be a long molecule of DNA that's going to be coiled together with several proteins. And this complex of DNA and the proteins and some of this RNA is going to be called chromatin. So the total genetic information that's carried in a cell or in an organism is called a genome. So we've done lots of studies in the past looking at genome-wide studies where we've sequenced basically all of the genes that we have in the human body. And we can use these genome-wide studies to evaluate differences across population groups, also to help to identify rare diseases or the causes of rare diseases. So there's a lot of work being done with the genome. It's very, very cool. That brings us then to the nucleolus. And this is my favorite of the organelles in the body. The nucleolus is a non-membrane-bound subnuclear body. So technically it doesn't really fall within the classification of an organelle, but it's close enough. And the main role of the nucleolus is going to be ribosome biogenesis. So It's primarily responsible for producing ribosomes. The nucleolus is going to be composed of 3 distinct subcompartments within it. We've got our fibrillar center, our dense fibrillar component, and our granular component or compartment. Each nucleolus is basically just going to be this kind of cluster of proteins and DNA and RNA. And so they're not going to be enclosed by any membrane like we've seen before. And so you can see here in these images, we've got our cell, we've got our cytoplasm and our big blue nucleus. And then within that nucleus, we've got these little green dots. Those are our nucleoli. Nucleoli, interestingly enough, are going to disappear and then sort of reappear during cell division. And they're going to reorganize into those new cells as they are formed. So, my work in graduate school focused on looking at the role of the nucleolus in triple-negative breast cancer. Now, you may have heard about the triple-negative breast cancer. It's a very aggressive type of breast cancer. It's very difficult to treat. So what I did was I looked at the number of nucleoli that are in each of the nuclei in different types of breast cancer. And I found that patients who had triple-negative breast cancer had more nucleoli per nucleus than what we saw in the non-triple-negative breast cancer tissues. So I thought about this for a while and came up with the idea that this is due in part because triple-negative breast cancer is very aggressive and it's has very rapid growth pattern. And so partly in order to keep up with this rapid growth, these types of tissues, this type of breast cancer needed additional or more nucleoli in order to be able to keep up with those growth demands. So that was my study. That was some of my work during graduate school. So here for your studying delights, we've got just a very handy table looking at all of the parts of the cell and their function, and this is also continued on this next table, on this next slide, because it is quite long. So, moving forward then, we're going to look at how proteins are synthesized by the cell. So much of the cellular machinery that's devoted to synthesizing large numbers of diverse proteins. So the cell devotes a lot of energy to making proteins. The proteins are going to determine the physical and chemical characteristics of the cells. So they're very important. And the instructions for this protein synthesis is going to be found in the DNA of the nucleus. And these proteins are going to be synthesized. This protein synthesis is going to involve both transcription and translation. So we'll talk about this in just a little bit with a little more detail. So first we have gene expression. So in a process called gene expression, a gene's DNA is going to be used as a template for synthesis of a specific protein. First, there's going to be a process called transcription, and the information that's encoded in a specific region of the DNA is going to be transcribed or copied to produce a specific molecule of RNA, ribonucleic acid. In our second process, then, we're going to transfer to a translation, and this is going to be when the RNA is going to attach to a ribosome, where the information that's contained in the RNA is going to be translated into a corresponding sequence of amino acids to form that new protein molecule. So this is a nice way of summing up the central dogma of all cell biology. It's very important. Is DNA is transcribed into RNA, and then RNA is translated into protein. All right, that is how it works. It's a it's a one-way street. It does not go backwards. So DNA makes RNA makes protein. Very important. All right, so that is the central dogma of cell biology. It's a very important concept. All right. So let's look at these processes just a little bit closer. Starting with transcription, and this is going to occur in the nucleus, and this is going to be the process by which genetic information is encoded in DNA, and this is going to be copied into a strand of RNA, and this is going to be used to then direct protein synthesis. So transcription is the process, like we said, by which genetic information that's encoded in DNA is copied onto a strand of RNA, And this RNA is called messenger RNA, or mRNA. And this is going to be what's actually going to ultimately direct our protein synthesis. So besides serving as the template for the synthesis of mRNA, DNA is also going to synthesize two other kinds of RNA. There's ribosomal RNA that's going to direct the production of ribosomes, and then there's transfer RNA, which we'll talk about here in a second. So the three types of RNA are going to be made up from our DNA template. Messenger RNA, like we said, is going to direct the synthesis of proteins. Ribosomal RNA, or rRNA, is going to join the ribosomal proteins to make up ribosomes. And then our transfer RNA, or tRNA. This is the RNA that's responsible for bringing in additional amino acids and utilizing its binding affinity with its anticodon region, which is going to interact with a corresponding region, or a codon, of that strand of DNA, right? Or a strand of RNA, excuse me. So, now then, looking at transcription. Transcription of DNA, continuing to look at transcription, I'm sorry. Transcription of DNA is going to be catalyzed by something called an RNA polymerase. RNA polymerases are going to be used, it's going to make use of a region of mRNA called a promoter. And this is basically like the big flashing light that says, hey, this is where you start synthesizing our new strand of RNA, right? Transcription of DNA strands is going to end at another special nucleotide sequence. This is called the terminator. So this is basically like our stop sign that tells our RNA polymerase when it needs to stop transcribing. So not all parts of the gene are actually going to be used to code parts of proteins. So for instance, regions within a gene that are called introns do not code for proteins or any part of the protein. So introns are going to be located between exons, and these are going to be parts of the DNA segment that do code for protein. So introns do not code for proteins, while exons do. Introns are typically excised during the whole transcription process. Next, for our process of protein synthesis, we have translation. This is going to occur outside the nucleus and is going to be the process of reading that mRNA that we've just produced, reading those mRNA nucleotide sequences to determine the amino acid sequence of a newly formed protein. So the whole process of translation is not too terribly complicated. So this translation is going to be simply the process of reading the mRNA nucleotide sequence to determine the amino acid sequence of that protein. So the sequence of translation is going to go about as follows. All right, for starters, the mRNA that's associated with the ribosome, or it's going to associate with that ribosome, and this is going to consist of tRNA and other proteins. All right, so here we've got our ribosomes coming together. We've got our tRNA in there to help to transfer on these amino acids based on the sequence for the mRNA. Specific amino acids are then going to be attached to the molecules of tRNA. And then another portion of the tRNA is going to have a triplet of nitrogenous bases that's called an anticodon. So a codon is going to be simply a segment of 3 bases of mRNA, and the anticodon is going to be the corresponding sequence of that. So if we have something that is let's see, what is it? if our mRNA is sequences, say AGU, right? Then we're going to have then our tRNA would have a TCA sequence there. So that's going to be our anticodon. It's going to match the opposite of the codon that we have, right? So the transfer RNA is then going to deliver a specific amino acid to the codon. And then the ribosomes is going to move along that mRNA strand as those amino acids are joined together to form our growing polypeptide. So here we've got our growing chain of amino acids to form that polypeptide. So we're not going to talk too much about DNA replication, but we're just going to mention a couple of things really quick. The first is that the 2 strands of the double helix are going to start by separating are going to be separated, and they're going to do this by breaking those hydrogen bonds that are holding our bases, our corresponding bases together, our A to our T, our C to our G, right? So those hydrogen bonds are going to be broken between the nucleotides. And then the new complementary nucleotides are going to attach to the proper site, and a new strand of DNA is going to be synthesized along each of the original strands. And so here you can see those little arrows are going to indicate the hydrogen bonds that are forming between those pairs of bases there. So now we need to talk about cell division, how a cell is going to be able to divide and make more cells. Cell division is the process by which cells are going to reproduce themselves. It's going to consist of a nuclear division step, either mitosis or meiosis, and then a cytoplasmic division. This is called cytokinesis. Cell division that results in an increase in bodily cells are called somatic cell division, and this is going to involve a nuclear division process called mitosis plus cytokinesis. And then cell division that results in the production of sperm or egg cells is called reproductive cell division, and it's going to consist of a nuclear division called meiosis and then also cytokinesis again. So cell division is going to be regulated by the cell cycle, and this is going to be an orderly sequence of events by which a cell duplicates its contents and divides in two. And this is going to consist of our interphase, right, which we can see here, and then our mitotic phase. So let's look at the process. It all starts with interphase. And during interphase, the cell is basically going to carry on all the various normal processes of life except for cell division. Interphase is going to consist of three phases. We've got G1, S, and G2. In G1 phase, the cell is going to be metabolically active, and it's going to be actively duplicating its organelles as well as any cytoplasmic components except for the DNA. Cells that remain in G1 for a very long time and perhaps will never go on to divide again are going to be called cells that are in G0 or G0 phase. So in the S phase then, chromosomes are going to be replicated, and then in G2 phase, cell growth is going to continue, and the cell is going to complete all of its preparations for cell division. The cell in interphase is going to show a distinct nucleus as well as the absence of chromosomes. Right? So after that happens, then we enter mitosis. This is the mitotic phase is going to consist of mitosis, which is our nuclear division, and our cytokinesis, and this is where the cytoplasm is going to divide. Mitosis occurs when the nucleus of the cell divides, and mitosis is going to result in the distribution of two sets of chromosomes into two separate nuclei. So mitosis is then going to be divided into four individual steps. We've got prophase, metaphase, anaphase, and telophase. So, for starters, in prophase, during this prophase, chromatin is going to condense into chromosomes, and the nuclear membrane is going to disappear, as well as the centrosomes are going to start to move to opposite poles. All right, so we can see that happening here. All right, next we have metaphase, and during metaphase, the centromeres are going to line up, and this is going to occur, this lining up of our chromosomes, the centromeres of our chromosomes are going to line up along that metaphyseal plate, or the metaphase plate. Next we have anaphase, and then during anaphase, centromeres of chromosomes are going to split, and those sister chromatids are going to move towards opposite poles of the cell. And then last but not least, we have telophase, and then during this phase, the mitotic spindles are going to dissolve, the chromosomes are going to regain their chromatid appearance, and the new nuclear membranes are going to form. That completes then the division of the nucleus, and so that all that's left is cytokinesis. And during cytokinesis, a cleavage furrow is going to form, so we're going to get this kind of dimple in the cell membrane, right? And eventually, the cytoplasm of the parent cells are going to fully split. So they're going to basically pinch inward until we've got two separate cells. So here we have a nice little table summarizing the events of somatic cell cycle for your studying convenience. And then moving forward, then we have to look at what's going to control whether or not a cell is going to continue to divide or is going to keep dividing, etc. There are three possible destinies for a cell. Right? The cell can remain alive and functioning, and it can never divide. The cell would be stuck in G0. The cell can grow and divide and continue with this pattern, or the cell could die. Enzymes called cyclin-dependent kinases, or CDKs, are going to regulate DNA replication, and turning these on or off, these enzymes, is going to be a function of proteins called cyclins. Right, that kind of makes sense. So the cyclins are going to control our cyclin-dependent kinases. Cell death, which is a process called apoptosis, is going to be triggered either from the outside of the cell or from the inside of a cell with a cell called something called a suicide gene or cell suicide gene that has been studied to a great deal. Necrosis then is something a little bit different than necrosis is something very different from apoptosis, however. So necrosis is going to be a pathological cell death due to some sort of injury. Right? So apoptosis is going to be just a natural occurrence in the cell's life. Once the cell gets to a certain age or is worn out, then it's going to undergo apoptosis and it's going to die. However, if you were to burn your arm or your skin really bad, right, those cells that were burned would undergo necrosis. And this is a pathological cell death, like we said, that's due to some sort of cellular injury. So, next up we have meiosis. And this is going to be the process that we also are going to call sexual reproduction. And this is where each new organism is going to be the result of the union of two different gametes. This is what we would call fertilization, and then one is going to be produced by each of the parents. So those two gametes. So in meiosis, the reproductive cell division that occurs is going to happen in the gonads. This is either the ovaries or the testes, depending on who you are, and this is going to produce gametes in which the number of chromosomes is going to be reduced by half. So as a result, the gametes are each going to contain a set of 23 chromosomes and are therefore going to be called haploid cells. We also can abbreviate this using just an N. If a cell has a full set of chromosomes, the whole 46 there, it's going to be a diploid cell. So fertilization is going to restore that diploid number of chromosomes. So again, we've got two haploid cells, two haploid gametes, one from each parent, coming together to restore our diploid number of chromosomes. Meiosis, unlike mitosis, which is going to be completed in a single round, is going to use two successive stages. We've got meiosis 1 and meiosis 2. So meiosis is going to need two stages in order to result in the production of more of these, the cell division of these sex cells. During the interphase that precedes meiosis 1, the chromosomes of the diploid cells are going to start to replicate. And as a result of this replication, each chromosome is going to consist of two sister cells, or sister chromatids. These are genetically identical. These are going to be attached at their centromeres. This replication of chromosomes is going to be similar to the one that we see that is going to precede mitosis in the somatic cell division process. So, now going on then and looking at the individual stages of meiosis, starting with meiosis 1. So in the first step here, the first of the two sister chromatid the two sister chromatids of each pair of homologous chromosomes are going to pair off, and this is an event that's called synapsis. And the resulting 4 chromatids are going to form a structure called a tetrad. All right. Then, during the formed tetrad parts of the sister chromatids of the homologous chromosomes is going to be traded. So this process is called crossing over, and this is what's going to allow this genetic recombination to occur. This is why siblings with the same parents don't look genetically identical, because we've kind of got this mishmashing here, this crossover process where different genes are going to be provided to different parts of the different two cells there. So this genetic recombination is going to be the result, cause those resulting sister chromatids to not be genetically identical. This is what's going to allow that genetic variability. So the net result is going to be a haploid cell with only one pair of homologous chromosomes, but with a paired sister chromatid. All right, that brings us then to step 2, and meiosis 2. Right, starting here, then the paired sister chromatids are going to make up each of those homologous chromosomes, and they're going to separate. The net result of meiosis 2 is going to be a haploid cell with one chromatid, and the net result of meiosis is going to be the production of 4 haploid cells, each of which is genetically different or genetically unique. And this is, again, because of that crossing over that we can see here. All right. So, in summary, meiosis 1 is going to begin with a diploid starting cell, and is going to end with 2 cells, each of which is going to have a haploid number of chromosomes. And then during meiosis 2, each of those 2 haploid cells that's formed during meiosis 1 is going to divide, and the net result is going to be 4 haploid gametes that are genetically different from our original diploid starting cell. So, with that, then we need to talk about aging in cells, how this happens. So aging is going to be just a normal process, just like everything else. Everything ages, it's just like so do cells. This is a normal process that's accompanied by a progressive alteration of the body's homeostatic adaptive responses. So the physiological signs of aging are going to be a gradual deterioration in function and capacity to respond to environmental stresses. So these signs are going to be related to the net decrease in number of the cells decrease in the number of the cells in the body, and to the remaining cells are going to function less efficiently, or going to have some cell dysfunction. The extracellular components of tissue, things like collagen, fibers, and elastin fibers, are also going to change with aging. This is why you get wrinkles as you age, because those collagen and elastin fibers that are going to make up the cell, or the components of that tissue, are going to become fewer or less effective. That brings us then to cancer. This is a group of diseases, and it varies widely, but this is overall is going to be characterized by uncontrolled cell proliferation. So cells that divide without control are going to develop into a tumor or a neoplasm. A cancerous neoplasm is called a malignant tumor or a malignancy. It's going to have the ability to undergo metastasis. And this is going to be where the cancerous cells are able to spread to other parts of the body. So you might have a tumor or a neoplasm, a malignant neoplasm, in one tissue of the body, and then it could undergo metastasis and spread those cancerous cells, to other parts of the body. So a benign tumor, something that's not going to cause problems, is a non-cancerous growth. So, there are several different types of cancer, and we name these, a lot of the times we name them based on the type of tissue that they arise from. So carcinomas are going to arise from epithelial cells. Melanomas are going to be cancerous growths in the melanocytes of the skin, those cells that are responsible for providing pigment to the skin. The sarcoma is going to arise from muscle tissues or connective tissues. Leukemia is a cancer of blood-forming organs. And then lymphoma is going to be a cancer of the lymphatic tissue. So, cancer cells, like we said, are able to divide rapidly and continuously. Normal cells in the body, they can only undergo cell division so many times before they're too old and they can't divide anymore. Cancer doesn't have that restriction. It just goes bonkers. It can divide and divide and divide. These cancer cells are also able to trigger angiogenesis. And this is basically triggering the growth of new networks of blood cells. So we've got these big clumps of cells, right, that are just growing out of control. And they're basically able to hijack the circulatory system and tell the body, hey, we need some more blood over here. So basically they just are going to stimulate the growth of new networks of blood vessels to feed this cancerous tumor. Cancer cells can also then, again, leave the site of origin, so they're leaving their original tumor, and they are able to travel to other tumors, or other tissues or other organs, and set up camp there. And like we mentioned, this process is called metastasis, and this is actually really the most dangerous part about cancer. Usually when people lose their battle to cancer, it's not, most of the time it is not due to the growth of the primary tumor. There are some exceptions, obviously, but usually it's the spread of this cancer, it's this metastasis that is what's ultimately going to be fatal in these cases. So, looking at causes of cancer, right? Lots of things can cause cancer, and it feels like every other day we're hearing something. Oh, this is cancer causing. Well, that's kind of true, and kind of that can be true. It's an unfortunate fact of life. So environmental agents are one of the things that can cause cancer to grow. So any kind of chemical agent or radiation that produces cancer is termed a carcinogen, or is said to be carcinogenic. And this is going to, in some way, induce some sort of mutation in the DNA. And this is part of what's going to allow those cells to divide unchecked. There are also viruses that can cause cancer. HPV, that human papillomavirus, is a sexually transmitted virus that is actually one of the major causes of cervical cancer. So there are also genes that can cause cancer, and these are known as oncogenes. So the normal counterparts to these oncogenes are called proto-oncogenes, and these are going to be found in every cell, and they carry out normal cellular functions until some sort of malignant change or some sort of crazy mutation in the DNA occurs to cause them to no longer be able to function properly. And some cancers may also be caused by genes called anti-oncogenes or tumor-suppressing genes. And these are genes that are going to potentially produce proteins that are normally going to oppose the actions of those oncogenes, or they're going to inhibit some sort of cell division. So they're putting the brakes on cell division. And if you, for some reason, mutate or lose that, mutate or make non-functional that gene that's putting the halt on cell division, then you're going to get this crazy out of control cell division that's going to lead to cancer, essentially. So lots of things can cause cancer. So it's good to know. So that is all that we have for today. So if there are any questions, comments, or concerns, as always, please feel free to email me or ask during class. So I will see you all next time.